Okay, in this video lecture, I'm going to go through chapter 17, which is titled Biotechnology, and point out a couple of emphasis things that you're going to need to know for your exam. So biotech has like really taken off in the last couple of years, or maybe the past decade, and it's incredible what we can do with genetics. So like right now, we can engineer DNA sequences by removing them from one organism, manipulate them, and insert them into a different organism. And right now, we do this a lot with genetically modified crops for increasing the quantity of food, and even like with animals. You can take a gene from a jellyfish, okay, the one that causes it to luminesce naturally, and you can insert it into this mouse, and now this mouse is going to express that gene product, or GFP, and it can illuminate. Okay, so it does raise some ethical concerns with what we can do with genes. So genetic engineering became possible after we discovered that bacteria enzymes could cut DNA at specific sites and basically cut the DNA that would allow other genes to be inserted into it. So think of it like a film strip. You cut the film strip and you separate it, and then you paste in a new film strip. So now it's like part of the movie. And we were able to do this, or we discovered this in the 1960s and 70s, and we've really like honed in on specific enzymes and how they can do this. So matching, mixing sequences from different sources created basically designer genes. And there's this awesome movie out there that kind of explores this designer genes. It's called Gattaca, Gattaca, play on the nucleotides. And I've always wanted to show it in college bio, but it does take a long time, and we're always crunched for time. But if you ever see the movie Gattaca, give it just watch it if you can. Okay, so since then, research has been concentrated on applying these techniques to use them for society. How can we take this information, okay, and solve problems like in medicine? With insulin. A lot of our insulin that we use to treat diabetes is actually from bacteria, because bacteria can produce it a lot faster. Before, we used to have to harvest it, from other mammals, and then like clean it and then package it, and that takes a long time. Well, with bacteria, mass production, they're fast to grow in a lab. You just insert the gene, they express it, they make it, and then we can just filter it and clean it and package it. Industry. Okay, and as well as agriculture, a lot with the genetically modified crops, pesticide resistant, drought resistant, everything resistant when you think about it. So the goals of genetic engineering is to improve our understanding of how genes work, and then advance that biotechnology, how when you manipulate an organism, hopefully you can create products that are useful or possibly cure a disease. So these are the learning objectives I'm going to go through, but I'm really going to talk a little bit about how it's done and tie it into a case study called pituitary dwarfism. So the pituitary gland produces a lot of very important molecules that stimulate growth, and there's specifically one molecule that we're going to look at, growth hormone, GH1 gene. So we're just going to look at one specific example. This molecule is 191 amino acids long. And what they've noticed is that people that have dwarfism, they have a faulty gene, okay? Defective copies of this GH1 gene. And so they're like, okay, so this gene is not making the hormone that it's supposed to be making. So could we treat the disease basically with injections of growth hormone, very similar to like treating type 1 diabetes? And it only worked if the hormone came from humans. They tried it with pigs, cows, and other animals, and it was ineffective. So in the 1980s, really the only source of this GH1 growth hormone was from cadavers. And that wasn't very effective, because you had to harvest 20,000 pituitaries to supply enough GH to treat a population of affected individuals. So the drug was rare and expensive, but that was the least of their troubles. I'm going to come back to that. I'm going to code to it right now. Why? Well, if the cadaver was contaminated with something called Creutzfeldt-Jakob disease, or Kuro, then the person that got that injection would also suffer the same fate. And you're like, okay, what is Creutzfeldt-Jakob disease, or Kuro? Well, these two diseases are caused by prions, and they are infectious proteins that cause degenerative brain disorders in mammals. And what they do is they go around and they cause normal human protein to become misfolded. And they just go around, and it's like almost like a zombie apocalypse in your brain. Meaning not that it's eating your brain, but it's creating holes, okay? These normal proteins, once they get touched by that prion, they become misfolded, and then they don't work right anymore. And they notice that people that have Creutzfeldt-Jakob disease or Kuro, when you examine their brain, it's literally sponge-like. And so Creutzfeldt-Jakob disease and Kuro is very common in societies where they practice cannibalism, okay? Because, when a person dies and if they have Creutzfeldt-Jakob disease, well, everyone, takes a bite of it, and then they end up with Creutzfeldt-Jakob disease or Kuro. So anyways, some children that were treated with the GH developed this disease, and so they decided to ban this in 1984. Okay, so when you see this like pink colored background slide, that is going to go back to this pituitary story gland. Okay, so basically they're like, you know what, we can't harvest cadavers. This isn't going to work. So we need to come up with a new game plan to possibly treat dwarfism if we wanted to treat dwarfism. Okay, so let's talk a little bit about how we manipulate DNA. So whenever you see the term recombinant DNA, it's basically a DNA molecule that's made from two different sources. Okay, so you have the gene from one organism and the DNA from another, and you're going to insert it, and it's going to become expressed. And, and then that organism is going to create that protein from that gene or whatever that polypeptide is, that enzyme, and then you can purify it and then hopefully use it for what you want to do. But recombinant DNA is a DNA molecule made from two different sources. And so how you go about cutting DNA to insert this gene is you need something called restriction enzymes. And what restriction enzymes do is they cleave DNA at specific sites. These restriction enzymes are actually called restriction endonucleases. And they cut DNA at specific sites, usually a palindrome, which I'll go into in a few slides here. So they were discovered in bacteria cells, and they noticed that bacteria cells had these restriction endonucleases that messed up the virus's DNA. So viruses, they infect the bacteria cell, injected their genetic material into the cell, but if the bacteria cell had restriction endonucleases, it would literally chop up the viral DNA, inactivating them, and the virus was harmless. Pretty cool. So basically the bacterial DNA was protected from being cut because of this enzyme that was chopping up a specific recognition site on the viral DNA and, preventing it from the virus being inserted into the bacterial DNA. So this was significant in two ways. It forms or allowed a form of mapping, physical mapping that wasn't possible because now you can cut DNA and map out where genes are. And then it also allowed us to create recombinant molecules because now we have the DNA cut naturally and we can insert new genes. So this is a diagram just showing how restriction endonucleases go about blocking it. But you can see that they have very specific sites that they would cut and they were digesting the viral DNA. So there are two types of restriction enzymes, type 1 and type 2 enzymes. And type 1 just means that they make simple cuts, but not at a recognition site. They kind of go about chopping it randomly. But type 2 enzymes, these are the ones that cut at palindromes. And when you have a palindromic sequence, it's read the same way in 5 to 3 on one side, as well as 5 to 3 on the other side. And so palindromic sequences are actually called sticky ends. And sticky ends just means that you can really splice together DNA very nicely. Okay, so remember, like a palindrome is like a word that is the same forward and backwards, like race car. So here is the restriction enzyme EcoRI, which is a very common one. I used it when I was at college, but you can see that it's creating a sticky end. TTAA, it's going to make the cut, okay? From this is one sample and this is the other sample of DNA. So it's going to make the cut so that way you can splice these two different DNAs together and create that recombinant DNA. So it just makes your life easier when you want to insert a gene if it cuts at a specific site. So DNA ligase allows the construction of that recombinant molecule, and those two ends of the DNA molecule are cut by that type 2 enzyme, and then they come together and DNA ligase pastes it together. So here's your foreign DNA, and you can see that it's chopped up into tiny little pieces, but it's chopped up in palindromes, okay? And then you have your host DNA chopped with palindromes, and then you put it together in a mixture, and you just hope that the enzyme DNA ligase will put it together, and you can do this with multiple genes. But it will splice or join the cohesive ends together, and now it is inserted into your DNA. Now, I'm going to talk a little bit about plasmids coming up here, but you'll see this term plasmids, and plasmids are basically vectors that carry the recombinant DNA, so I'll touch base on that. That is definitely something you're going to need to know for your exam, but like I said, I'll get there. Okay, so here's something you're definitely going to need to know for your exam. It's called gel electrophoresis. And what a gel electrophoresis does is it separates the DNA based on size, its charge, and some other properties as well. And how it works is you have this gel, okay, this gelatinous gel, and areas called wells that you put in your DNA samples. And then you're going to run a current through this. Now remember, DNA has a charge, okay? It is negative, and opposites will attract. So when you run this current, it will go from negative to positive. Literally, DNA will move through this gel and create bands. Okay, so you would run it for a period of time, and then from there you will look to see how far the bands travel. And depending on how far they travel, that rate is inversely proportional to its size. Okay, so if it travels a far distance or a long distance, then I believe the fragments are short. Okay, so here is an example of DNA, and you can see that it's cut palindromic here. Actually, I probably should have slid that over better. But, and it's broken up into 3 segments. We'll just call it W, the segment X, and the segment Y. And when it runs from negative to positive, so it's running down the well in this direction, negative to positive, you can see that my Y is short, and it ran the furthest, and then X, and then W. Z and Y in comparison to Y, because Y is over here. Okay, so I have 2 strands. I'm sorry, I'm thinking out loud. I haven't lectured on this in over a year. And then here's Z. Okay, so I have 1, 2, 3, 4, 5. Okay, so these are my 2 samples. So this is one sample, and this is the other sample. So Z is a very long fragment, and it doesn't go very far because it's big. But Y is short on both ends, and it's the furthest. Okay, so what you need to know about gels is that we do this like in CSI and CIS. They run gels to compare DNA samples with a suspect to the actual evidence. And basically, if the bands match with the suspect, so in this case, like a crime, then you have your criminal or the perpetrator or whoever you're testing this against. So you can see that these bands do not match up. These are from 2 different samples. If the bands did match up, I could say, oh, if this is the evidence from the crime scene, and this is the suspect. And if the bands matched up, I could say, there's a pretty good chance that you were at the crime scene. That is your DNA. All right, so that's kind of how gel electrophoresis works. Okay, so still continuing on with bacteria and viruses. Remember, viruses, when they hijack bacteria cells, they insert their DNA and basically hijacked the host cell. So we're kind of using the same thing with this idea of taking this recombinant plasmid and injecting it into the E. coli host cell. So this plasmid is a vector. It carries the wanted gene you want expressed, and if we insert it into the host cell, it will, take in that DNA and start to read that gene. And now it's been transformed. So transformation is cells that take up DNA from their environments, and they do it all the time, and then they incorporate it into their own genomes. So we can propagate this process and create a transgenic organism. Okay, wow, that was a lot. Moving on to molecular cloning. Okay, so a vector is something that carries the recombinant DNA, the gene that you want, and then the host cell. And there's 2 types of vectors that are used, plasmids and phages. And phages are viruses. So you can use a phage. But we tend to use plasmids, which are small, circular extrachromosome DNAs that are dispensable to the bacterial cell. So guarantee exam question, something about what do plasmids do, or what are vectors? I don't know, something like that. But vectors and plasmids, they go together. They help carry the gene that you want expressed to, that organism and then become incorporated. So plasmid vectors are clone small pieces of DNA. You'll take your vector, you'll isolate it. You have your bacterial plasmid, you have your gene of interest, and you somehow want to get them together. And so to get them together, you're going to use the restriction endonuclease. So it cuts at specific sites, a palindrome site, so that they stick together. So you're going to create some sticky ends, then you're going to hope DNA ligase pastes them together, and then you're going to culture it in a lab and hope that the bacteria takes it and expresses that gene. So then you'll introduce the clone vector into the cells, you'll clone your cells, and then you will do your best to identify which cells took the gene of interest. Now, phage vectors are larger than the plasmids. So if you have a very large vector that you want to insert into, then you're going to use a phage, okay? So bacteriophage lambda affects E. coli. You'll isolate the vector, you'll package it inside the head of the virus, and then put it with E. coli so it can infect E. coli and insert it in the same process. So just a diagram just showing how you would use a phage, inject it, and so on. Okay, so with it, when you start to insert genes and culture it, basically what you're doing is you're creating a genomic library. And so a DNA library is just a collection of DNA fragments that have been cloned into vectors. There's two types of libraries, genomic and then the cDNA, which is made with your clone reverse transcribed. It's cloned into plasmids. These guys are the vectors. These are the large fragments of DNA. Do you need to know this for your exam? No. Okay, so this is a breakdown of plasmid library. That would be your cDNA, which is smaller. And then over here is your phage DNA, which would be your genomic. But like I said, you don't really need to know that. With the cDNA, it stands for complementary DNA. And the reason why it's complementary is because you're going from mRNA using reverse transcriptase, which is something you'll need to know for your exam, to create into a DNA strand. So usually information flows from DNA to RNA to a protein, but now we are going backwards. We're going from mRNA back to DNA through an enzyme called reverse transcriptase. So the benefit of using a cDNA is that you don't have any introns to worry about because you're just going straight from mRNA to DNA. When you're going from DNA to RNA, you usually have to edit or modify it, okay? But that's, like I said, not very important unless you're going to go into this field. Okay, so back to the pituitary gland. So what researchers did is they isolated mRNAs from the cells in the pituitary gland, and they used reverse transcriptase to create the DNA. Now, to go, to do this in a human, to go from DNA to RNA to a protein, it's how do we know which introns are spliced together? Like, it would be a very long process. So it's easier to go from mRNA back to DNA and just forget the introns. Let's just have all the exons ready to go. And so that's exactly what they did. So we went from mRNA back to DNA. So now we've created a complementary DNA library, and then put it into restriction endonuclease, chop up the sticky ends, place the gene that we want into a vector and create some recombinant plasmids, and then we're going to grow it. So how do scientists find the sequence of interest in a random collection of DNA fragments? You got to do something called molecular hybridization. Like, how do you know which E. coli or which colony of bacteria is expressing the genes that you want? Well, usually we will probe or tag the gene of interest, okay? So we are going to take advantage of how DNA unzips. And basically DNA denatures or separates from each other when you reassociate it with the correct complementary strand, or you heat it up to get it started. So the known strand that you know is called a probe, and it will bind to the complementary sequence when it's being analyzed. So this probe, you have to label it some way, either with a radioactive atom, like phosphorus or sulfur or some type of fluorescent dye like GFP, so you can see it illuminate in the colony. So radioactive DNA will bind to that complementary sequence in your cDNA. And this is kind of how it works. So here's your probe labeling, okay? And then you'll denature it, pull it apart so that it matches complementary to where it should be. And then, you'll put it in your colony, and then it will actually like light up, okay? And then you're like, okay, that's the gene of interest. It has the gene that we want, and go from there. So we use the genetic code to infer approximately the DNA sequence of that GH, that growth hormone 1 gene. And it was possible because we knew the amino acid sequence. Okay, I think I told you it was like 191 amino acids long. We knew the sequence. So the genetic code is redundant, so it was approximate. Because remember, there are many codons that can code for the same amino acids. So it's just a little bit of guesswork. So then we were able to synthesize many copies of the short single-stranded DNA that was complementary to the inferred approximate sequence. And then we used a probe to identify which cell contained that, I inversed it, growth hormone, all right? And then once it was embedded, hey, now we can mass produce it, okay? Insert it into a plasmid, put it in E. coli, let E. coli do its thing, transcribe the gene. There are no introns. Everything is expressed. There's no splicing required. We're not messing around with that, which is awesome. And then all of a sudden, the human growth hormone accumulated in cells, and we isolated it and purified it. All right. DNA analysis. So there's a few different ways that you can analyze DNA, not just by gel electrophoresis, but something called blotting. And there's 3 types of blotting that I'm going to go through. So southern blotting reveals DNA differences. Basically, you're going to take DNA, cleave it up into tiny little fragments with a restriction endonuclease that's separated by a gel electrophoresis. And then it's blotted by a sheet of paper over the top, and it will transfer some of the DNA strands to the sheet. And then you're going to filter and incubate it and label it with some radioactive probes, and you're going to get a single strand of DNA for that specific gene. So when do you use southern blotting? If you have a DNA sample from an individual and wish to find a specific gene in that sample, you use southern blotting, okay? The other 2 blots, so original, a northern blot and a western blot. So the northern blot's the same, but you're using mRNA instead of DNA as your starting material. And then a western blot is similar, except that you're going to separate proteins via the electrophoresis and then probe it and use antibodies to bind to the protein of interest. Okay, so if you're wondering where the names all come from, it's a lighthearted tribute to the protocol where it was all produced and derived from. Edwin Southern started it, and then everyone just kind of played off on the other variations of it. So, okay. You can trace inheritable patterns and identify specific mutations using something called restriction fragment length polymorphism. The procedure is, again, you're going to use a restriction enzyme that's added to DNA. It's going to be incubated for several hours. You're going to use a restriction enzyme to cut it at those recognition sites. You run it through a gel. You get fragments according to size, and then you can visualize whether or not it was cut by the enzyme. But again, don't need to know that unless you're going to go into this field. You can sequence the DNA, and that can tell you some information about the genes and genomes. So I think there's 2 ways you can sequence it. One is enzymatic sequencing, where you're going to use dideoxynucleotides as chain terminators. And this one, it's kind of tedious, but you're going to perform 4 separate reactions, each with all the A's, with all the C's, with all the T's, with all the G's. And then you get fragments, and it's going to be separated by a gel. So here you can see the 4 separate reactions, okay? And so here's the C, here's the T, here's the A, here's the G. So to read it, if I'm going from here to here, I'd be like, okay, the A band is first, okay, then it goes T, then it goes, oh, there's another T. That's not how I'm reading that. I feel like it should be A, T, A, but you basically read the bands in order as you go down. So here's the G. Oh, because it's complementary. God, I'm dumb. Okay, so A, T, T. Okay, I get this now. C, A, G. So you just go in order, but it's complementary. So then here is the sequence right here. Okay. You can still do that, but now we have automated sequences, which means we can use machines and fluorescent dyes, and it reads it a hell of a lot faster. So it's using a photo detector, which is not pictured anywhere. You can also analyze DNA using a PCR. You can take a single sequence and amplify it for analysis. So if you have a sample that's like too small and you want more of it, then you're going to use a PCR. So what you do is you denature the DNA so it unzips, and then you're going to add primers. Basically, the DNA is cooled, and in these primers, you have a bunch of nucleotides, and it basically does DNA replication in a nutshell. You have to make sure the temperature is right. You have to use a specific polymerase that can be used at these high temperatures, usually a TAC DNA, which is found in a, what is it called? An archaea. So remember, there's 3 domains of life: eukarya, bacteria, and archaea. And archaea live in extreme environments. So this TAC DNA polymerase is found in archaea that can live in like hot pools, like in Yellowstone or something. But anyways, it basically does DNA replication in the lab. So you get a large enough sample to analyze. Okay, moving on to genetic engineering. So when vectors are expressed, you can make gene products. It works because the sequences are there, they are expressed in the DNA, the bacteria are transformed, they start to produce large amounts of protein. Like I said, we're doing this right now with insulin. Okay, the insulin that we use to treat diabetes is coming from bacteria. You can also take genes and you can introduce them across species lines, right? Different barriers. You can take genes from E. coli and insert it into a plant. And so when you take it from different species, we call it a transgenic animal because the gene has been introduced without the use of conventional breeding. So, you can take the eyeless gene from a mouse and insert it into a fruit fly and cause a substitution in the Drosophila gene if you really wanted to. Cloned genes can be used to construct something called knockout mice. And a knockout mice is where a gene is inactivated and you want to study the effect of it or loss of function, and you basically insert it into that organism to see the effects of it. Okay, so you no longer have to chemically induce or radiate the species to cause the mutation. If you know the gene and you want to cause a loss of function of that gene, that's where this comes into play, the in vitro mutagenesis. So how do you engineer a knockout mouse? You take embryonic stem cells from a mouse, and then you take a look at the DNA. You want to introduce that DNA that contains the mutant gene. So I don't know what that mutant gene is. Maybe it's the production of black fur. I don't know. But you insert it into the blastocyst stage of a brown mouse, and you let it do its thing. And you implant it into the surrogate, and then the surrogate mother gives birth to a chimeric pup. And this pup has cells from both the black strand and the brown strand. Okay, so then it's a knockout because it's not expressing the cells that it's supposed, or the genes that it's supposed to be. So some medical applications of biotech, all of this information, is human proteins can be produced in bacteria. That means it's cheap. We can do it in bulk and create a large amount of proteins. Okay, we do it with arterial peptides. This is used to treat high blood pressure. You just use it for tissue plasmogen activator, which is a protein that causes blood clots to dissolve. So if someone has a stroke and they get this TPA, which is made by bacteria, and it's used within 3 hours, they might be able to regain a lot of the mobility or loss that they would have if they didn't have the TPA. You can also simplify vaccine production. Okay, so the DNA is extracted from a virus. You have the gene isolated, and then you purify it and basically make it less harmful by using the surface markers and the proteins and just creating a vaccine. So for example, I have a slide here that talks about the production of the HB vaccine, which is hepatitis. So here's the hepatitis B virus, and we understand the antigen, the thing that produces the antigen. So we take it out, we put it in a plasmid, paste it together. Notice this came from a virus, this came from bacteria. So now we have our recombinant DNA because it comes from two different DNA sources. And then we're going to put it in a yeast cell that is eukaryotic. And the yeast cell is going to create the HB antigen that we can take and purify and extract, and then we package it into the vaccine. It's kind of cool. Gene therapy. I mentioned this in class a few lectures ago, but it can treat genetic diseases directly. If you have a hereditary disorder because of a faulty gene, this would obviously be a way to cure it by adding a working copy of the gene. Now, obviously, this is controversial, and it has some successes as well as some recent failures. So there are some that you can treat with gene therapy. People are being treated by using gene therapy. They're basically trying to create a working copy of the gene. But like I said, it has some successes and some failures. So it's not 100%. And I think a lot of it might have to do with the splicing of the mRNA, the introns, and all of that. It also has agriculture applications, which are actually pretty common. Crops are modified to resist disease, tolerate herbicides, change the nutritional value, which I'm going to talk about on the next slide. It's so cool. Produce pharmaceuticals and other biologically active compounds. So one case here is, you can take a tumor-inducing plasmid from plant bacterium, and you can transfer genes into a broadleaf plant. Like you can literally take one gene from one organism and put it in the other. You can take crops resistant to herbicide, glyphosate, and create no-till planting, so you're not losing soil to soil erosion. Oh, and also just like increasing the health of the soil, because if you till the soil a lot, the bacteria in the soil, which is like good, if you till it a lot, then you have lower numbers and lower diversity. Bt toxin, pest-resistant crops. Now, pests don't eat it, but it can cause damage to like wildlife, pollinators that we really want. So there's a downside with that one. Golden rice. This one is the one where you can change nutritional value. So people in Africa, okay, they don't have a lot of vitamin A in their diet. Okay, vitamin A is like in vegetables and fruits that are orange. Okay, so like carrots. Okay, yeah, that's why they say eat your carrots, because it's good for your eyes because of vitamin A. And vitamin A is, is needed for vision. So in Africa, people were suffering from, diseases of the eye. And they're like, oh, it's because a lot of them have vitamin A deficiency. So how can we help that? Well, they actually genetically modified rice and inserted a gene for the rice to create a higher concentration of this provitamin A, and they call it golden rice. And so now in areas where there was golden rice, they saw lower levels of eye diseases. So it's kind of a cool thing. Genetically modified crops do raise a number of social issues. The safety. Genetically modified crops haven't been around long enough to study long-term health effects. There is a movement of possibly taking these genes and introducing it to their wild relatives. I always tell the story about how like salmon fisheries or farms, they are usually genetically modified, like ones that are kept in farms. So they're like bigger and I don't know if I want to say meatier, but they're just bigger and bigger for the fillets, right? And I remember reading a news article like years ago in college how like some salmon escaped from the fishery and are out in the wild, and they were concerned about them breeding with the wild, and would they beat up on the other salmon because of their size, or be viewed as freaks? I don't know. It's just kind of an interesting article to read. I wish I could find it, which could obviously lead to possible loss of biodiversity. Okay. Biofarming is using transgenic plants to produce pharmaceuticals, such as maybe serum, some other subunit vaccines, and some antibodies. We have found that we are more successful with transgenic plant technologies than we are with transgenic animal technologies. So utilizing it in plants has a higher success rate than it does in animals. Okay, so back to the pituitary. So there are some ethical concerns over the recombinant growth hormone. GH supplies increased. Physicians used it for treating not only people with dwarfism, but to children who were just short, and they were not considered dwarfism. And so the treatment required several injections per week, and it actually increased their height by a few centimeters. So now the drug was not just being used to treat dwarfism, but it was being used cosmetically. And so the question is, if short people are discriminated against in a culture, is a medical treatment a better solution than just education and changes in our attitudes? So that does it for the biotech portion of this. So the two things you really need to pull away is make sure you know what a plasmid is, and make sure you can read a gel electrophoresis, and understand that if the bands line up, then they are, from the same species or the same sample. And if they don't, then you are dealing with different species. Okay. All right. Thank you.